Hello friends, today we are going to learn about introduction of blood and the properties of blood as a fluid. It is a part of Veterinary Physiology 001, Ali's Veterinary Wisdom. So, regarding introduction, we know that blood is a fluid connective tissue and it consists of cells and other cell-like formed elements suspended in fluid called plasma. Main function of blood is to carry oxygen or gases, nutrient molecules, and wastes. The blood volume is typically values given as a percentage of body weight are 7 to 9%. Now, come to the functions of the blood. Blood helps in transportation of oxygen, carbon dioxide, nutrients, hormones, and waste products. It regulates and maintains homeostasis, acid-base balance, body temperature, water balance, and also regulates osmotic pressure. It provides immunity. Properties of blood. There are 6 properties: hematocrit, blood color, blood volume, blood pH, specific gravity, and viscosity. Now come to hematocrit. HCT is internationally recognized abbreviation for hematocrit. Proportion of cells to plasma is the general definition of hematocrit. Clinically useful measure of pathological conditions, and technically we can say that when a column of blood is centrifuged, the components are separated according to their relative specific gravity, and the cellular components occupy the lower portion and taken together are known as hematocrit, while plasma occupies the top portion and it is the liquid component of the blood. And you can see it in the following picture. You can see hematocrit in normal condition, which is the volume of the blood cells, is around 45%. Below 40% it is anemia, and above 50% it is polycythemia. Now come to the other property of the blood, that is blood color. We all know that red color of blood is due to the presence of hemoglobin within the erythrocytes. Gradations of color from bright red to bluish purple are seen depending on their degree of saturation of hemoglobin with oxygen. The greater the saturation, the brighter the red color. Plasma is yellow to colorless depending upon the quantity and species examined. The color is due to yellow is due to the bilirubin. Bilirubin is a product of hemoglobin degradation. Now in different species we can see what is the color of plasma, like in cats and dogs. Further we can say small ruminants like sheep. And goats. The plasma is either colorless or very slight yellowish in color. While in case of cows, the cattle, it is generally the dark yellow in color. The plasma is a dark yellow in the cows. Further, in horses, it is even darker than cows because of relatively high bilirubin content in the plasma of horses. Now, come to the other property, blood volume. Blood volume is a function of lean body weight, and it is generally 8 to 10% of body weight, which we have already discussed. Blood volume cannot be measured directly because exsanguination, removal of blood from body, results in the loss of only about 50% of the blood. The remainder is trapped in the capillaries, venous sinuses, and other vessels. Erythrocytes volume and plasma volume can be measured by various techniques. If one or the other is measured and the hematocrit is known, then we can measure the blood. Other component is the blood pH. The normal pH of the blood is 7.4. The venous blood is slightly towards the acidic side because of the increased content of the carbon dioxide which form the carbonic acid, whereas the arterial blood is slightly more towards the alkaline side. Plasma is more alkaline than the blood corpuscles. Blood pH range. In case of dogs, it is 7.32 to 7.68. In case of cattle, the cows, it is 7.35 to 7.50. In case of horses, it is 7.35 to 7.43. And in case of chicken, that is fowl, it is around 7.56 to 7.65. Another property which we are going to discuss is specific gravity. Specific gravity is the ratio of a given volume of a fluid to weight of the same volume of distilled water measured at 25 degrees Celsius. Plasma protein concentration is largely responsible for the specific gravity of plasma. The cellular elements called the corpuscles have higher specific gravity than plasma. And copper sulfate drop falling method is commonly used for the assessment of the specific gravity of blood and plasma. Now the conditions in which the specific gravity falls or increase. First of all, specific gravities decrease in the following conditions like pregnancy due to increase in plasma volume, anemia, renal diseases because loss of albumin, edema, the hemodilution and starvation and malnutrition. Specific gravity is increased in following conditions like polycythemia, severe dehydration and hemoconcentration. The last but not the least, the viscosity of blood. Viscosity of blood is influenced by the concentration of RBCs and plasma protein. Among plasma protein, the viscosity is mainly contributed by gamma globulins. It is important, which provide the resistance to the blood flow and helps the normal pumping activity of heart. Viscosity of blood is determined using viscosimeter. An increase in the viscosity of blood is seen in conditions like polycythemia, congestive heart failure, jaundice, vomiting, diarrhea, etc., whereas decrease in the viscosity is commonly noticed in anemia and edema. One more thing which is important, that normally the viscosity of blood is around 5 times greater than water. Thank you for watching this video. This is developed by and presented by ExoneVet under name Ali's Veterinary Wisdom. If you like it, please subscribe our channel ExoneVet on YouTube. Thank you.